Welcome to the Scientific Sense podcast, where we explore emerging ideas from science, policy, economics, and technology. My name is Gil Eapen. We talk with world's leading academics and experts about their recent research or general areas of topical interest. Scientific Sense is an unstructured conversation with no agenda or preparation. We cover a wide variety of domains where new discoveries are made and new technologies are developed on a daily basis. We are most interested in how new ideas affect society and help educate the world how to pursue a rewarding and enjoyable life rooted in science, logic, and information. We seek knowledge without boundaries or constraints and provide unedited content of conversations with researchers and leaders who love what they do. A companion blog to this podcast can be found at scientificsense.com, and this podcast is available on over a dozen platforms and directly at scientificsense.net. If you have suggestions for topics, guests, and other ideas, please send them to info@scientificsense.com, and I can be reached at gil@eapen.info. My guest today is Professor Peter Rogan, who is Professor of Biochemistry and Biostatistics at Western University and Professor of Oncology at the University of Western Ontario. His lab focuses on bioinformatic technologies at population scale to address important biomedical challenges in genomics and other areas. Welcome, Peter.
Thank you.
I want to start with your recent work in geostatistical analysis in the context of COVID positive cases in the United States, in which you say geostatistics analyzes and predicts the values associated with spatial temporal phenomena, and it's a practical means of describing spatial patterns and interpolating values for locations where samples were not taken, and measures uncertainty of those values, which is critical to inform decision-making. You want to talk a bit about that work?
Sure. So just to put some perspective on this, geostatistics has been around for at least 50 years. And it really has been pioneered and championed by the natural resources extraction industries. So for those of you who aren't aware, if you want to find a deposit, whether it's oil or mineral, usually they do soundings with ground radar, and the reflection of that sounding reflects what's down underneath the ground. And basically they map these reflections. They sample different locations, and from those samplings, they can build a map and basically determine where to dig or drill, as the case may be. And so you can think of this from an operational point of view as, substantially reducing the amount of effort involved in finding the deposit that you're interested in. So the way it works is that you have several measurements, and the expectation is that the measurements, when they're closely spaced together, will be correlated with one another. As far as you move further away from the deposit, then the sounding will change and you expect to see a different measurement. So the idea then is that if you have these measurements, you can sort of infer what's between them. That's called interpolation, spatial interpolation. Now, also you can do this by looking at, for example, ecosystems. And so you can, for example, look at pollution in water over time. So there's not only a spatial component, but there's a time element to this.
Right.
You can see how the system changes when you're measuring something and interpolating what's happening between the points that you measure. Okay, so with that as kind of the background, imagine that you have counts of patients with COVID-19 in every state in the United States. And in fact, the famous Johns Hopkins web server coronavirus resource
Yeah.
publishes, as of the 25th of March of this year, counts in every county of every state in the United States.
Right.
So if we apply the same principles of geostatistics, then we expect that where there are hotspots, those counts will be increasing, and that won't be occurring in areas where the virus is either under control or it's not being transmitted because people aren't going there or congregating there. So you expect to see a lack of correlation, spatial correlation. So we have different geostatistical tests that we can do to actually find those hotspots.
Right. Yeah.
Go ahead, sorry.
Yeah. So I can understand that, Peter. So at the beginning of this issue, there were only few cases. So if I understand this correctly, Peter, let's say take a state like Texas, you would take a few measurements, and from those measurements, both in space and time, you can have some expectations of how that could change. And using that information, you could actually potentially predict how the spread is going to be, something along those lines.
Well, right? It's possible, but we're not doing any prediction. Right?
Okay.
We're currently just, first of all, we're not just taking a few spaces.
Yeah.
We download the entire data set for the entire United States when we do the analysis. And we're essentially, we analyze it different ways. So for example, you can look at distances. So you can say, I want to consider each county and then compare it with every other county that's within 50 miles of that county, mm-hmm. right? Mm-hmm. And so now you have these pairwise comparisons, and you're essentially looking for whether the counts in one county are different from the other ones around it.
Right.
All right. Okay. And that is a statistically significant result. Now, let's say I do that analysis and I do it over a period of a week.
Yeah.
And I see the same result. Well, what that tells me is that the virus is not under control. And it's also not noise, right? So we're seeing a signal. We're seeing that signal consistently over the course of a whole week. And we're not looking at a noisy result, something that's just a fluke that might happen on a particular day because the counts happen to be higher, because the testing laboratory happened to be more efficient that day, or any of the other possible, there could be errors in the Johns Hopkins data, which we've come across. There's all kinds of reasons why on a given day you might see a signal that you might not see the next day. So by doing the space-time analysis, we get a more reliable, more consistent result.
Yeah, so what exactly is the output? So is it some sort
of
infection?
So yeah, I'm happy to show it to you, but it's not so easy. I think I've given you the...
Yeah, so could you describe that? So, from a policy...
I'll do my best. So there are two types of geostatistical analyses. One is called area-to-area analysis. So that's like comparing the centers of different counties with one another, but recognizing that the number that we measure represents all the patients in that county, all the individuals who are affected in that county. So we're comparing counties with one another. And then there's this other technique, which most people have probably never heard of, but that's really the basis for the natural resource extraction methods, and that's called kriging. And there we are actually, for example, we know exactly where the individuals who were affected are. Now, of course, that information isn't reported publicly, but it is available to some people, and so you can actually use what's called linear regression, ways to infer and estimate the levels of COVID in like individual neighborhoods, for example, or maybe even on individual streets, when you have the locations of the individuals within that county. So that's called point-to-point geostatistics. So what we're, with regard to the area-to-area analysis, we're getting what's called a confidence measure. So we can statistically generate what's known as a p-value, a probability that this is something that we might see at random, right? And if the probability that it's a random distribution of cases is high, then we can't exclude that it's that it's a real hotspot. But if that probability is non-random, that is, the likelihood of seeing, counts in one county is so much higher than seeing the counts in the adjacent ones, then we can exclude the so-called null hypothesis, the hypothesis that this is a random distribution of individuals. And hence, that means that the individuals you see that are clustered in that particular county came about by transmission of the disease from different people localized in the same area.
Okay, so from a decision-making perspective, from a policy perspective, what this will provide us, you are still looking at data backward and essentially saying, if I look at, area XX or X1, what I'm seeing in that area, it's not a statistical fluke. It appears to have some real sort of infection intensity, or whatever term you want to use it. And so whoever is responsible for that county from a policy perspective could utilize that information. Is that the utility?
So there are two really interesting results, one of which is incredibly practical.
Yeah.
So when we started doing this, what has kind of kept me going on this was we decided to explore the case of Louisiana first. So everyone knows that in the first Tuesday of February in Louisiana, there is a very big party, and it's called Mardi Gras. And people come from all over the world to attend that party on Bourbon Street in New Orleans. So when the government realized that, COVID had arrived on the shores of the United States in January, and I guess sometimes in February, the first New England Journal article of a patient in Seattle was in late January, as I recall. Anyway, New England Journal of Medicine article. So it was expected that Mardi Gras would cause a hotspot. And of course, we know that the incubation time is somewhere in the vicinity of 2 weeks. And so there was an expectation that after Mardi Gras, at least some of the individuals who had migrated to New Orleans stayed there, or some of them were locals, and that we would expect to see a hotspot. So we thought that would be a good choice for our first test. And lo and behold, literally on the first day that Johns Hopkins started to report data, we saw a hotspot in New Orleans and in a couple of the surrounding counties, and they persisted for quite a while, actually. But what surprised us was that we also saw a smaller signal, one in northwest Louisiana in Shreveport, as well as in the Baton Rouge area, but that one was a bit more transient. The Shreveport signal came around for us on the 25th of March. So fairly early, but then it kind of disappeared and then it reappeared. But the interesting thing was that, we're in a position when we look retrospectively to see when the media as well as governments reported these case counts being increasing on the internet and the newspapers and so on. And the first reported case for Shreveport, the first reported announcement occurred on April 1st. So the point was that the geostatistics told us 6 days before the government announced it that they had a problem. Now, imagine you were a health department for the state of Louisiana and you knew you had 6 days of advance notice. Well, there's a lot of things you can do. You can start to isolate people. You can start to institute distance, constraints that prevent people from congregating, and you could potentially prevent the spread of the virus.
Right. Yeah, I can see that, Peter, when at the beginning of a pandemic. But I'm wondering, when you have a situation where you have 70,000 new infections per day, do you still get some utility from it?
Well, that's no. So I wanted to talk about that too. So in our most recent release of the archive, we decided that we would explore the question of what happened after, I mean, which we all know about, but I just thought we would see it on a spatial level.
Yeah.
What happened after Memorial Day when the president and others decided to relax the distance constraints and told everybody to go back to work and play and so on. And you can see in some locations, especially Texas, the spread of these hotspots, literally 2 to 4 weeks after. One of my technologists said that, and I don't remember the location, but there was, after this was noticed by the government authorities, they actually instituted the mask requirements in that particular location, and then those hotspots started to diminish in that particular location. So it's one way to measure effectiveness, but I decided to do one more thing with these data, where we were looking, in Southern California and Texas and Florida. We decided to go all the way back to the beginning of our analysis, and we actually saw some hotspots that were transient in each of those places, and then they disappeared. When social distancing was mandated and everybody was in their homes for the months of March and part of April, I guess, and part of May, and then of course when you see the disease reappear after the relaxation of the constraints, the interesting thing was you don't see it reappearing elsewhere where there weren't early hotspots.
Right.
So what that tells us, and we haven't yet published this, but we're writing it up now, is that it looks like there's a hidden reservoir of virus. We know that there are individuals who are infectious for months on end, right? And they have symptoms. We also know that something like 30 to 50%, depending on your age group, is asymptomatic.
Right.
And it's also known that those asymptomatic individuals produce as much virus as the symptomatic ones do.
Yeah.
So it's very reasonable to suspect that when the lockdown occurred after the end of March, early April, that we had these carriers, if you want to call them that, who were asymptomatic, walking around, and that all that was necessary was for the constraints to be released, then everybody went to the bars and the transmission occurred, and we see what we see today in California, Arizona, Texas, and Georgia and Florida, and not to mention elsewhere. So I think, to me, there's really only two solutions to this. And I mean, I'm not an epidemiologist, but I have a long history of doing molecular diagnostics, including an infectious disease in my former company.
Yeah.
And there's really only two solutions. One is random testing everywhere, right, which the government and the United States are not equipped to do. And the other is a home test. So basically, the same way you'd have a pregnancy test, you'd have to have, maybe you might even need to buy a device, but basically people would need to have it in their homes. And you would need to basically certify that you actually were COVID-free before you could go to certain places. Yeah. But bottom line is we need to ramp up testing dramatically if, in fact, there are hidden reservoirs of virus that are not apparent, because they will always continue to recur and resurrect, right, when we do relax these constraints.
Yeah, so that's very interesting, Peter. So, would you say, given that there is a high prevalence of infection that is asymptomatic, what we might see is sort of, different waves of this thing happening. So one could argue that you need some sort of a dynamic policy that needs to kick in very quickly, something based on analysis like yours that picks up that wave early on and very quickly adopt a lockdown or a close to a lockdown policy that is local. But it sounds to me, correct me if I'm wrong, it sounds to me that policy has to be dynamic, and this might be going on for a while.
Well, it's certainly likely to me that, first of all, it's not just that it's dynamic, but the asymptomatic individuals have to have a persistent infection. So I would suspect the people who are being called super spreaders are not just asymptomatic and positive, but they're asymptomatic and they're carrying it for months.
I see. Oh.
And I can't imagine that every asymptomatic individual is testing positive for more than, it's a limited number of people. We need to identify who those are. And the challenge, of course, especially given the rattling about freedom and so on, is to get somebody who doesn't have symptoms to be mandatorily tested.
Right.
That would be very difficult in the United States, I think. You'd have to mandate that everybody gets tested, or you have to mandate that there's random testing.
Yeah, there is, as you know, there is a thought that if you increase testing, you will pick up more infections, and hence reducing testing might be a good idea.
It's a terrible idea because the problem is that the people who are infected at this point in time, we don't know which ones are going to progress to the intensive care unit and die. Now, that was what my other paper about that I sent you.
Yep.
Was, and we can talk about that if you're interested.
Yeah. So let's jump into. I don't know which one you're referring to. I was going to jump into another piece of work that you did very recently, and that is interpretation of mRNA splicing mutations in genetic disease, review of the literature and guidelines for information theoretical
analysis.
Well,
I only provided that to you as background, right? So
yeah, could we talk a little bit about it? I find that very interesting, Peter. So
okay, but I think the reason I sent it to you is that this COVID hypothesis that we have for why some patients get very sick, is it uses some of the technology in that splicing paper, right? So mRNA splicing, our mRNA are expressed genes, right? So we have genes in our chromosomes, and then we make copies of those, and they're expressed. And then we have the central dogma of molecular biology, and those expressed genes get translated into proteins, which are, the structural elements of many cells. The question is, some of these mRNAs, right? They actually they're processed. So one of the differences, it's actually really not so much of a difference, but humans and eukaryotes, animals, plants, we have interrupted genes. Mm-hmm. So the parts of the genes that get turned into proteins are separated from each other. And they're separated by sequences in the RNA that are ultimately in the DNA, and those sequences have to be spliced out.
Right.
And that happens for something like 97% of all human genes. Mm-hmm. And some genes have many segments that have to be spliced out, like the dystrophin gene that causes muscular dystrophy when it's mutated has, it's the largest gene in our genomes, and it has several hundred segments that need to be spliced out precisely. And hence, some of the mutations that cause muscular dystrophy, are affected by that process going awry.
Right.
Well, the whole process of recognizing the sequence that has to be spliced out is carried out by proteins.
Yeah.
Right. And in particular, they're carried out by a unit in our cells called spliceosomes. Mm-hmm. That recognize both ends of the segment that gets removed, and then brings together the segments that ultimately get translated by the ribosome into forming proteins, the actual proteins themselves.
Right.
So this splicing process involves proteins which we call RNA binding proteins. Mm-hmm. Right? They're very important proteins for maturing the RNA that's transcribed from genes into the mature processed mRNAs, messenger RNAs that get translated into proteins. So these RNA binding proteins, there are actually many thousands of them.
Yeah.
And the question is, how do you recognize sequences in different genes? They're different sequences, all of which encode these signals that say cut here, right? Splice and cut here. And so I've been working in this area, I don't know, since the mid-1990s, and we use a technique known as Shannon information theory, and Shannon was a very famous man in the 20th century. He devised and wrote some key studies called the mathematical theory of communication when he was at Bell Laboratories.
Mm-hmm.
And it is the reason why we're able to communicate right now, right? Because we can compress data and we can encode data and we can encrypt data thanks to Shannon's mathematical theory of communication.
Yeah.
But the thing that's interesting about it is that Shannon information theory applies to any kind of information, not just electrons passing through integrated circuits, but also sequences, DNA sequences, RNA sequences, right?
Mm-hmm.
Protein sequences. And so you can look for signals in DNA and RNA and proteins that are, and quantify those signals in the same units that we quantify the information in our computers. So when we talk about, a megabyte or a gigabyte, those are made up of bits, right? And a bit is a choice. So choice is it's either 1 or 0. In a transistor, it's 1 or 0. So in DNA, we have 4 different nucleotides.
Yeah.
So there's 2 different choices. Okay, so every position in DNA has 2 bits of information.
Right. Yeah, I want to, sort of, so you can see that information spectrum, so to speak, and is that useful in making some sort of prediction?
Yes, it is.
Yeah.
But more importantly, it's actually the thing that's remarkable about information theory is it's actually related to thermodynamics.
Mm-hmm.
So we can actually directly relate the bits of information in a site that's recognized for splicing to the energy that's required to recognize that site.
Yeah.
Okay. And there's a formal proof and a theorem about this. And so that is what brings it back to biochemistry, right?
Mm-hmm.
So we can have strong binding sites, we can have weak binding sites. When we have mutations that affect the splicing process, they weaken a binding site.
Yeah.
Often, right? They also can create brand new binding sites, right, when there are mutations that in the wrong locations. So the splicing event doesn't actually occur where it's supposed to.
Yeah.
Okay. So the thing about these information theory models and software and algorithms is that they're very general, right? We can derive a recognition sequence for any RNA binding protein. Mm-hmm. Even for any transcription factor. We've done that too, okay? Right. That controls DNA, that activates or represses a gene. So it simply takes a series of binding sites whose sequence we know for a particular binding protein, and then we derive this mathematical model, which is an information weight matrix that allows us to distinguish the sequences that are recognized from all the other sequences in the genome or the transcriptome. Okay?
Right. And so
now can we come back to COVID? I'd like to actually bring it back to the COVID story.
Yeah, exactly.
So the virus that causes COVID is made out of RNA.
Right.
And the paper that I sent you, which was published about a week ago, we put forward this idea that COVID basically can kill lung cells. And the question is, how does it do that?
Yeah.
And it can kill lung cells very rapidly. We actually calculate about 2 days is what it would take for it to happen. Mm. So if you infect a cell with a single virion, a single viral particle, and it starts to copy itself, replicate itself, because it encodes its own replicase, its own polymerase to copy its own genome. It can make enough copies in 2 days to cause the cell to blow up, to lyse. We use the term apoptosis.
Yeah.
The way that this occurs actually is that the virus contains these same binding sites that are also found in our cells' genomes. Okay. So when produce a brand new RNA in the nucleus of our cell by transcribing it. Before it gets turned into messenger RNA, there are RNA binding proteins that bind to it and protect it.
Right.
And by protecting it, it prevents that RNA from basically forming a duplex, a double strand, a double helix with the remaining DNA strand in the chromosome.
Mm-hmm.
So if it forms that double helix, we get something called an R loop. So there's one strand that's not bound to anything, and the other strand is bound to RNA. And something your readers, your viewers might not know, listeners might not know, is that when you form a duplex between RNA and DNA, it's actually stronger than the duplex between DNA and DNA. Okay, so it's a big problem, because if we have these things form, then what happens when the chromosome replicates, the normal chromosome replicates, is it hits these loops, these R loops, and it causes chromosome breaks because it can't progress. We can't copy the chromosome.
Right.
And these R loops basically then are damaging, and we activate, DNA repair processes, like when we're exposed to ultraviolet light or radiation or anything else, carcinogens. The problem is that the virus is continuing to copy itself and produce binding sites for these RNA binding proteins, which can no longer find their way to the nucleus and protect the normal host RNAs.
Now, why is this different, Peter, from the typical RNA virus infection?
It isn't different.
It isn't different.
Fact,
the same thing is happening.
Actually, in our paper, we talk about, I found some evidence, published evidence, on influenza and dengue, which are both replicate in the nucleus. I know influenza does. I can't remember where dengue virus replicates. Right. But as you well know, there are a small proportion of patients who have influenza infections who die for the same basic reasons as the patients with COVID-19. Basically, they end up in the intensive care unit. They can no longer transport oxygen across the alveolar surface into the bloodstream. And so they develop multi-organ system failure because their outlying tissues are starved of oxygen and they die, right? So the thing about this hypothesis, right, is that the virus is basically causing all this damage all by itself.
Yeah.
Now, I'm not saying that the immune system doesn't play a role, but you can appreciate that when the virus starts to replicate, it's doing so inside the cell and there's nothing stopping it, right? So if the virus has a jackpot replication event in a particular cell, there's nothing the immune system can do to prevent this. There's the so-called innate immunity, which is basically signals that cells send out to tell the immune system that they've been infected, and that sends macrophages and cytokines to the location of the infection.
Yeah.
The thing is that cold viruses and coronaviruses, MERS, for example, they actually encode proteins in their viral genomes that downregulate, that repress this signaling system. So the immune system is unaware that there's an infection.
Right.
Okay. And so the virus can replicate unimpeded as these RNA binding proteins are synthesized in the ribosome. Instead of being transported to the nucleus and protecting host RNAs, they can get bound by the viral genomes.
So
in depleting these RNA binding proteins, it allows the R loops to form. There are so many of them that they overwhelm the DNA damage repair systems. And the cell basically gives up and sends signals to commit suicide. That's apoptosis. The problem with that is you've got all this virus that's waiting to escape, and it all gets released all at once.
Yeah, so the question would be, if this is happening in any sort of RNA-based virus issue, so number one, the mortality rate for COVID is a lot higher, number one. Number two, we see a very high variability among individuals, right?
Yeah.
Some having very severe
Yes.
symptoms and some asymptomatic almost.
Yes.
So how do you explain that in this
Well,
case?
first of all, there's no evidence, for example, that dengue and flu virus encode proteins that prevent innate immunity,
Yeah.
right? So it's very possible that the immune system, as it does, often takes over and gets rid of those cells, and so you recover. But it's still a very serious killer in the United States
Mm.
every year, right? So what was the other point?
No. So
Well, that's one of the issues is that that coronaviruses in general, and this one in particular. The other issue is that you rely on the fact that there are these jackpot events, right? So you can imagine in some cells, maybe virus replication doesn't occur quite so quickly. Maybe in some individuals, their immune system is actually educated by regular common cold viruses, and they do, and maybe the vaccines will actually get rid of the majority of virus that gets into our systems. But the thing about this mechanism we're proposing is you only need one viral particle to infect the cell, and for that infection to cause a jackpot replication event. And the reason why remdesivir works so well is not just because it's preventing replication of the virus, right? So it's an analog, I believe it is of adenosine,
Mhm.
and it prevents replication of the virus. But if you can imagine, if you don't replicate the virus, you can't produce these RNA binding sites that the RNA binding proteins need to bind to impede normal host RNA processing. So that's how I believe remdesivir basically makes patients better, including the ones that are in the intensive care unit.
Right. So, but if I understand this correctly, Peter, you said the immune system can't really take care of it because all this stuff is happening inside the cell. And often the immune system is not even aware that infection has happened, right? And by the time the issue has happened and it has replicated, it is sending a large number of replicated RNA inside the body. It's probably too late for the immune system to do anything. If that is the case, again, why do we see a big difference between the typical flu and COVID?
I don't have an answer to your question, right?
Yeah.
But I guess I would like to talk about something else that's in the paper
Sure.
that's sort of relevant.
Yeah.
And that is, we all are aware that there are age-related differences in susceptibility to this virus,
Right.
right?
Right.
So if you're older, like me, you don't go out because, you're more susceptible, right?
Yeah.
And if you're a child, well, the probability of dying and getting a severe infection is lower, but there are still children who are in the intensive care unit. Why would children show some protection? So the answer, I believe, and I think there's actually some articles to support this,
Yeah.
is that the lung cells that are infected are these so-called AT2 cells. They're type 2 pneumocytes. Now, it's possible they also infect type 1 pneumocytes.
Mm-hmm.
So the difference between type 1 and type 2 pneumocytes is type 1 pneumocytes actually transport oxygen across the alveolus into the red blood cells, right?
Yeah.
Which we need. Type 2 cells are like helper cells. They're a bit like astrocytes in the brain. So they're necessary to help us breathe. They make something called surfactant. And you may recall that premature babies get surfactant.
Mm-hmm.
So surfactant is basically this mixture of proteins and lipids that allows your lungs to expand and contract easily without, causing tissue damage.
Yeah.
Right. It's kind of like bathing your lungs in sort of an oily substance that allows it to move, right? So So you need the most amount of surfactant when you are born, when you
Right.
take your first breath, right? So babies who are premature, because surfactant is made very late in gestation,
Yeah.
need supplementary surfactants. So there's synthetic surfactants, and it actually saves many lives, many neonates.
Hmm.
So if you look at the production and the lifetime of these type 2 cells that make surfactant, you're born with the maximum number of them.
Yeah.
And as we grow older, we lose them.
Right.
So if you think about the fact that this is the cell that gets infected, if we have a jackpot COVID replication event, then that jackpot event, once it releases virus, it doesn't have to infect. There aren't as many cells that it needs to infect in order to cause its damage. You're already substantially compromised because you have fewer of these infectable cells, these susceptible cells, right? Because you're older,
Right.
right? And so I think we can't really say, what's the probability that a particular virus is going to cause this. But here's the thing: if you look at virology textbooks, they will all show, usually they'll use flu as an example, but sometimes they'll use other viruses. They'll show an electron micrograph of virus being released from the membrane of the cell, right? In a normal sort of process. The cell doesn't die. It doesn't undergo apoptosis. It just releases this viral particle, right? So that level of release of virus is something that I believe that the immune system can handle.
Right.
But when you literally blow up the cell and release a large amount of virus into the alveolus, if there are not a lot of type 2 pneumocytes, those cells have a higher probability of infecting the remaining pneumocytes and completely compromising function. So, you hear these stories of people who arrive at the hospital and they're sick, but within a few hours, they no longer, they're on ECMO, they're on extracorporeal membrane oxygen machines, right? It's not enough to put them on ventilators.
Yeah, it
Ventilators
progresses.
aren't going to solve this problem, because if your cells that transport oxygen are dead, no matter how much oxygen you force into their lungs, it's not going to solve the problem.
Right. So then would it be correct in saying, Peter, that in the case of COVID, what you're saying is that the mechanism is slightly different. It is not, getting a cell infected and then replicating in there and slowly getting back into other cells, but rather when the first cell get infected, there is sort of an explosion that happens there that
In
releases
some
a very
people,
large.
where these jackpot replication events are occurring, and so
Right.
I think in some people, for whatever reason, it's maybe stochastic. I'm not sure. I mean, obviously, I don't have all the answers. We're still only a few months into this.
Yeah.
But I've made some proposals to the Canadian government. Hopefully, at some point, they'll consider it. Consider them. But basically, the idea is that the individuals that are rapidly progressing and ending up in intensive care units are releasing large amounts of virus into their bloodstreams, and that may be why they have these infections for many months.
Right. And so what are the implications for vaccine then? I know that there are different methods
So
being tried, right?
it's an interesting question. So, in theory, the vaccines, they induce these neutralizing antibodies, right? So let's say you're infected, but not by a large amount of virus. Well, if you have a strong vaccine response and you make a lot of neutralizing antibodies, it could be that the virus is eaten up by macrophages, or by T cells and B cells, and basically cleaned out of your bloodstream or your respiratory tract before it can cause serious damage. So it may well be that some of these vaccines are very effective in some people. I think there's another point, though, and that is that because the virus has the ability to disguise itself, there's never been an effective vaccine for the common cold.
Right.
People have been working on it for many years. The second most common cause of the common cold is a coronavirus.
Mm-hmm. Mm-hmm.
Okay. So why is that? It's because of this ability that coronaviruses have to repress innate immunity. So it may very well be that we need to get booster shots every 3 months. With tetanus, we get a booster shot every 10 years. I mean, so it may well be that we may need to get booster shots of the vaccine on a regular basis.
Yeah. I mean, there was, there was a hypothesis that said, people who are able to manage the infection are those who have had several coronavirus-like attacks in the past. And if your hypothesis is right, that could be true as well, right?
Well, that's another point about children, right? Is they always have a cold, right?
Yeah.
So they're already making antibodies. That could be another reason they're protected.
Yeah, but if that's the case, then there is a question around the resident time, because an older individual presumably have had many, many more infections than a young individual, right? And hence, if the resident time of the antibody is long enough, presumably the older individual is more protected. But that is not what we are seeing.
Well, again, there is this fundamental problem of the MERS virus makes a protein. I don't even remember what it was called. It's in our manuscript, but it's well documented that it represses the interferon response. And the interferon response is this very primordial, this innate immunity, primordial response to viruses in particular, because viruses make something that are ordinarily not made in our cells. They make double-stranded RNA.
Yeah.
So when the virus replicates, the next thing it does is it has to make its own proteins. And coronavirus basically has this fairly complex mechanism for shifting the strand from a leader strand from one strand to the other to begin to initiate replication of the negative strand of the virus, which encodes the proteins that it co-opts our ribosomes to actually make. Mm-hmm. So the virus, as part of its life cycle, makes structures that have two RNA strands in them. And that's what the interferon response recognizes. Mm-hmm. Influenza virus does this as well. Dengue virus does this as well. So RNA viruses activate this very primordial response. And the problem is that coronavirus makes a protein that shuts down the interferon expression. Hmm. So if it does that, right, then it's the chances that the virus can, basically sulk and evade the immune system is much higher. Right.
Yeah. So you mentioned remdesivir, and there is some evidence that it appears to be effective. So what can we learn from that? So I'm going back to this idea that the different types of vaccines being tried. There are, I think, 6 into phase 3 now, and I think there are at least 3 different types of vaccines. Do you have a view as to which vaccine is more likely to succeed?
It's a hard question. So there's a lot of noise and a lot of excitement about the Moderna vaccine, which is mRNA based. So you don't have this classical, let's inject the antigen into a chicken egg and make the antibodies. And it's a much simpler approach. You'd literally make the RNA, you let the cells in the body make the RNA. I think it's interesting, it's attractive, but there is this concern, right? Because you put a lot of synthetic RNA, which has been chemically modified to avoid nucleases inside cells and inside the body to break down that RNA. But that RNA that it's making is basically encoding this spike protein that the coronavirus makes, right? It encodes part of the spike protein. Well, it's going to contain some of these RNA binding protein binding sites.
Yeah.
So I don't know whether or not that might cause a cross-reactivity, or it might affect. I don't know. I mean, if a person doesn't mount a substantial immune response after receiving the Moderna vaccine, the question I have is, what's the fate of that RNA, and could it be interfering with host nuclear mRNA, RNA metabolism? I don't know.
Right.
I think that the details of these different vaccines have not been really released, right? Exactly what sequences they're using as immunogens and so on.
Right.
So it's not obvious at this point which vaccines will work. I suspect that there could be ethnic specific differences.
Mm-hmm.
There could be, one thing that's clear is that there's going to be a marketplace for these vaccines. And the physicians who are prescribing them and using them, there will be a lot of optimization that goes on to try to figure out what algorithm should be used, which vaccine should we use on which individuals first, second, third, and so on. And there, I think perhaps the genome sequences of these individuals may turn out to be helpful in helping figure out who's likely to respond to which vaccine. I don't know.
Yeah, so, just like any other viral infection, the more the viral load is, the higher the chance of getting severe infection and problems. But if your hypothesis is right, Peter, if I understand this correctly, that is true. But there is another dimension here, which is some people, even with very little viral load, could get into a situation that, whatever you described in terms of sort of an explosion happening and very quickly going down. The initial infection load was very little, but that doesn't really correlate with what actually happens to that individual. But we don't really know why, right? Who and what is causing that.
So all I can say is that this concept of a jackpot replication event is definitely going to require a lot of study, right? But the people who have a jackpot replication event are certainly, so, coming back to information theory, we have made mathematical information theory based models of two different proteins that bind to host RNAs. And they also bind in multiple locations to the flu, the dengue, and of course the COVID, the SARS coronavirus 2 RNA sequences. And it's really simply the law of mass action. If you replicate large amounts of viral RNA, then you will soak up, you will deplete, you will sponge up lots of these proteins. And what do these proteins do? So about 10 years ago, there was some really seminal papers out of Rockefeller University where they downregulated these two proteins that we study here. And they showed that, first of all, the SRSF1, which is actually a splicing factor, but it was originally, in my view, and probably in view of the Rockefeller University scientist, Dr. Manley and his colleagues, its original purpose was to protect the RNAs from forming these R loops.
Yeah.
And we essentially made a mathematical model based on 50,000 of these binding sites, and then we scanned the host transcriptome. We also scanned the viral transcriptomes, and we figured out how many binding sites needed to be bound in the viral sites, how much replication needs to occur in order to basically soak up half of all of the SRSF1, right? So at some point you hit this threshold whereby the RNAs in the nucleus aren't protected enough anymore and the explosion occurs, right? The virus is replicated enough times that it soaks up this essential protein. So Dr. Manley's experiments 10 years ago showed that when he synthetically down-regulated, he repressed this particular protein, cells basically started to undergo apoptosis. They started to lyse. They died.
Yeah.
was by this process that is this suicidal process, because they couldn't repair the damage that occurred
Right.
when the replication forks hit the R loops. There were just too much damage.
Yeah.
Then a couple of years later, they found another protein that when added back to the cells, it's RNPS1, it actually complemented, and that's a genetic term, it recovered, it rescued the deficiency in SRSF1, right?
Mm-hmm.
So what we found when we actually built our models of these binding sites is they're very similar to one another. They actually overlap one another in like 70% of the cases in the human genome.
Right. I think you mentioned this.
That's what's actually, I think, going on, right? We've quantified what the RNA binding proteins are, where they bind in the virus, where they bind in the host, and how much virus you need in order to prevent the binding in the host.
Yeah, so very quickly, I think you mentioned this, Peter, but I just want to touch on this again. So the remdesivir effectiveness, does it give us any clues at all?
Yes, right. Remdesivir, essentially, so when you polymerize, when you make a copy of the virus, right, you're essentially copying the adenosine, guanine, uracil, let's see, and adenine, right? You make copies, right? You're duplicating the viral genome, right?
Mm-hmm.
So the remdesivir is a modification of adenosine.
Okay.
And when it gets incorporated into the viral genome, it prevents any further nucleotides from being copied. Mm-hmm. So it's what we call a chain terminating nucleotide, right? So chain termination, we've known about this for a long time, right? It's the basis of how we do DNA sequencing.
Yeah.
So if the virus can't make copies of itself, right, it can't kill cells.
Yeah.
But because in order to kill cells, it has to have copies of itself to bind to these RNA binding proteins.
Yeah, that is true with any sort of infection. I was wondering if it gives us any clues. I think it is effective in severe infections, if I understand it correctly. So I was just wondering if there is anything that tells us, what is causing that explosion that you mentioned to be dampened by the drug. But
well,
I think
the drug basically prevents the virus from continuing to replicate.
Yeah.
So if the virus can't continue to replicate, then maybe your type 2 pneumocytes don't die. Right. So you have certain number of those pneumocytes in your lungs. They can start to replicate themselves without virus impinging on their own replication, and potentially you can recover. Right. Okay. So you can appreciate that because we understand the mechanism of action to some degree of remdesivir, there are a lot of drug companies working on other nucleoside analogs that do the same thing as remdesivir, that cause chain termination, right?
Right.
The other thing that's interesting as a potential therapy is, and I talk about it briefly in the manuscript, and I even cite an article of another drug.
Yes.
So in order for transcription of the virus proteins to occur, right? The RNAs that encode the virus proteins, like making the various proteins that make up the virus and that encapsulate the RNA. There is this template switch that occurs from the positive strand after it's replicated to the other strand, to the negative strand, and then that initial event of transcribing the viral genome to make proteins involves another host protein called hnRNPA1, which has a completely different function in the nucleus during splicing. But it seems to be important for initiating expression of viral proteins from the viral genome. So there are, in fact, inhibitors of hnRNPA1 that could be tried as therapies to prevent the viral proteins from being synthesized.
Yeah. So in conclusion, if you were to handicap, the likelihood of us getting an effective vaccine in the next 6 months to 1 year, what would you say to this?
I'd say it's high.
It's high. Okay.
I think that, as I said, the challenge is going to be defining effective, right?
Yeah.
Medicine always starts out in a fairly blunt kind of approach, right? So we have a control group who doesn't get the vaccine, and we have a group that does get the vaccine, right? And the thing is, that group that gets the vaccine, they're not stratified in any way, right? You have to make a decision. Are we going to give this to older individuals? Or are we going to give it to younger individuals? You could make the argument that you should give it to younger individuals because as we get older, our immune systems start to senesce, and they don't work so well to actually battle viral infections and other sorts of infectious agents, bacterial infections.
Yeah.
So the chances of the vaccine actually being effective will be better in individuals who have strong immune systems to begin with. But of course, that's not the population that we need to protect, right? So how you decide who you're going to test the vaccine on, how you're going to stratify, what are the eligibility conditions? You should look really close at that when they talk about the approval of a vaccine. Who were they tested on? Right. So if you're a person who's 65 years old and you're not that well, you'd really want to know that the vaccine's effective in other 65-year-olds that might have a acute respiratory distress, adult respiratory distress syndrome, or some other precondition that might make you at higher risk to die of this virus. So I think that's a key question people should ask when vaccines become available, because there'll be a bunch of them, right? But how the patient population was selected is going to be important.
Yeah, I mean, the Moderna phase 1 trial, there was a lot of excitement coming out of that, and obviously that is not the target population that is just looking for safety there. So I think that's a good point. The question would be, when we prove this is effective, whatever it is, who is the target population it was tried on, and how does it sort of generalize to the population? That is one question. The second question is, what is needed at the societal level from a herd immunity perspective? Perhaps there are different strategies one could play. If we can get to herd immunity without exposing the high-risk patients, maybe there's another way to tackle the problem. I don't know. But yeah, there are a lot of questions.
I just want you to consider something else. I don't know any details about this, but I read somewhere that Gilead is testing remdesivir as a nasal spray.
Yes, I saw that, yeah.
Right? Well, delivery, right now it's delivered in the intensive care unit with an IV drip, right? But if you could go to the drugstore down the street and buy a nasal spray, when you felt a cold coming on, you don't know what the cold is, but what if you could potentially prevent the viral infection from expanding in your body early, right?
Yeah, that'd be a big deal.
So I actually, when we first started thinking about this hypothesis, and I started reading about coronaviruses, and I'm not a virologist, but as we've kind of talked about today, what you want is to take what SARS-CoV-2, what COVID-19 is, and turn it back into the common cold.
Yes.
Right? So if there was a way that we could use a medication that would essentially stop the virus cold early, so that we never develop these severe symptoms that would potentially kill us, then it really is no different from a common cold.
Yeah, that's a great way to think about it, Peter. Excellent. Yeah, this has been great, Peter. Thanks so much for spending time with me, and good luck with all the research that you're doing in this area.
Well, thank you. Well,